till in previous class we have discussed about the role of XXL gene. So XXL gene is present in both male and female. In case of female, XXL gene expression is controlled early by the early promoter and it leads to the formation of XXL early protein. So this XXL early protein will help in the splicing of XXL protein, which is formed with the which is transcribed or which is formed later on, and it is transcribed with the help of maintenance promoter. Maintenance promoter is present in both male and female. Okay, but the early protein, the early XXL gene product, that is XXL protein, early XXL protein is only present in females, not in males. Now, this early XXL protein will help in the splicing of the protein which is formed by maintenance promoter. Maintenance promoter of XXL gene will also form XXL protein, but the splicing of the RNA, which is formed by which is formed with the help of the maintenance promoter. The maintenance promoter will help in the transcription of XXL gene, and it will lead to the formation of XXL mRNA. So this XXL pre-mRNA, this XXL pre-mRNA will undergo alternative splicing. So what happens in alternative splicing, and who will help the alternative splicing? Alternative splicing is held by early XXL protein. So early XXL protein is helping in the alternative splicing of maintenance promoter the RNA which is formed by the maintenance with the help of maintenance promoter. So what will happen in the alternative splicing? So in the XXL RNA, XXL pre-mRNA consists of 8 exons. 1, 2, 3, 4, 5, 6, 7, 8. When these so what happens in case of female, the third exon, this third exon is chopped by early XXL protein which is only present in female. So when this third exon is removed, so in total we'll have 7 exon: 1, 2, 3, 4, 5, 6, 7. Okay, so that is 1, 2, 3, 4, 5, 6, 7. So now we have 7 exons. These 7 exons, if we see 1, 2, now because 3 is removed, so 1, 2, 4, 5, 6, 7, 8. In total there are 7. The RNA, this mature RNA will lead to formation of XXL protein. This XXL protein, remember, is formed with the help of maintenance promoter, and this RNA, the RNA which you are seeing here, is formed because of the help of maintenance promoter. The alternative splicing in this XXL mRNA is held by the XXL pre-XXL protein which was formed by early XXL which was found by the early XXL promoter. Now, what is there in this third exon? So why this is removed? So if you see the male embryo, what happens is that here we have the 8 exons. Third exon is not removed. Third exon is not removed, and here we have 8 exons. Basically, this third exon has a stop codon. Because of presence of this stop codon, the transcription, so the translation process will not be complete. The translation will not be complete. There will be a non-functional polypeptide because of premature termination of translation. Whereas the third exon was not there, and so complete XXL protein was formed in female embryo, which is XX embryo. Now, why the XXL protein expression is so important? Why XXL, we know the name is sex lethal gene. Why the expression of sex lethal gene is deciding? Why the expression of sex lethal gene is so much important? Now we'll see it. So here you can see that heading is differential regulation of transformer gene. So transformer gene is now a very important gene. So we write transformer gene as TRA gene. So this TRA gene expression is basically dependent on XXL protein. Now XXL protein is a protein which is formed in female embryo, and XXL protein is not formed in male embryo. Now, because the XXL protein is formed in female embryo, and we know that from this line, the first line says that the expression of TRA gene is controlled by XXL protein. So this means that the TRA gene expression is dependent on XXL protein. So when XXL protein is there, it will lead to formation of TRA protein. Okay, so it is clear that the TRA protein is going to be formed only in females because it is controlled by what? XXL. It is controlled by what? XXL protein. Now the second line suggests that the TRA gene is transcribed into pre-TRA mRNA. The TRA pre-mRNA is spliced alternatively to create a female-specific mRNA and a non-specific mRNA. So if this is a RNA, so this is a RNA. So this is spliced in a way. This is spliced in a way. So there is a TRA RNA which will further form a which will further lead to formation of TRA protein, and there is one non-specific mRNA. So here also alternative splicing is going on. So what you have to remember is that the TRA pre-mRNA which was found is basically alternatively spliced with the help of XXL protein, and which lead to formation of TRA mRNA and a non-specific RNA. So male What happens in male? A male-specific tRNA contains a termination codon in the second exon. So we remember. So basically, you remember that the third exon of XXL protein was consisting the termination. The third exon of male XXL RNA was having a stop codon, which lead to the premature termination of translation in male embryo, and thereby there was no XXL protein. So the male-specific tRNA contains a termination codon in second exon. Okay, so here, because the XXL protein is not present, the termination. So XXL protein is not present. TRA-mRNA will form. TRA-mRNA will form in male, but because a second exon, which is consisting of stop codon, will not get removed because of absence of XXL protein. Thereby, the TRA protein will not form in male. It will only form in female because the XXL protein is only present in female, and this XXL protein will lead to the alternative splicing of pre-TRA mRNA, and this will lead to the removal of and this will lead to the formation of two mature RNA. First is trimRNA and a non-specific RNA. TrimRNA will form the TRA protein. So the word is transformer gene. We will see that why the name is transformer gene, and why it is so much important, because it is going to decide the fate of an embryo further. Now, this second exon is not utilized in female-specific mRNA. Female-specific tRNA is translated into functional TRA protein, same as of XXL protein. But in this figure, it is clear that the TRA RNA was not, the second exon was not removed, and because of this, there was no protein in case of male. Whereas the XXL protein, which was present in female, this lead to the alternative splicing of this TRA. So what you can see, the alternative splicing of this TRA RNA, and that you can see now this RNA is short. Only the gray part is there. Here you can see a red part is also there, which is consisting of here what you can see, a red part is present in case of male. Because of this, there was a premature termination of translation, thereby there was no protein. Here you can see the XXL is helping in the alternative splicing. Because of this, red part is gone, and you can see that TRA protein was formed. So what I want to repeat is again that So we read about the sex lethal gene. Sex lethal gene is present in both male and female. So the sex lethal gene consists of two promoters, a early promoter and a maintenance promoter. Early promoter helps in the transcription of early the early promoter help in the transcription of XXL gene, which lead to the early XXL protein, and it is only present in female. Whereas the maintenance promoter helps in the maintenance promoter present in both males and female. The maintenance promoter is present in both male and female. So the maintenance promoter will help in the transcription of XXL gene in both male and female. But the RNA which is formed in male is the RNA, the pre-mRNA which is formed in male, the third exon will remain there, and the third exon has a stop codon because of which premature termination of translation will happen, and there will no formation of XXL protein. Whereas in case of the female, the early XXL protein, which was there with the help of the early promoter, help in the alternative splicing of the RNA of the pre-mRNA of XXL gene, and because of this, the third exon is removed in female, and a mature XXL XXL protein is formed in case of female. This XXL protein will help in the alternative splicing of a transgene mRNA, and because the XXL protein was not present in male, so there will be no alternative splicing of the second exon, and this second exon will present there, and it will lead to the early termination of translation in case of male, whereas in case of the female there is XXL protein which will lead to the alternative splicing of the tRNA, and the TRA protein will form. So till now, the XXL protein and TRA protein are present in female, whereas XXL protein and TRA protein are not present in case. Now there is a third gene. So now third gene is coming into picture, which is double sex. That is the double sex gene is an autosomal gene. The double sex gene is an autosomal gene that can produce two different proteins through alternative splicing of its mRNA. So one thing is clear, that through alternative splicing we can create new proteins. So I am guessing, and I know that you have read about alternative splicing in your class 12 also. A gene name called TRA2. So I want to clarify here again that in previous slide you read about previous slide you read about TRA gene, not about TRA2. So I'm talking about TRA2 gene now. So second line suggests that a gene named TRA2 produce TRA2 protein both in male and female. So again, so the XXL protein and TRA protein are present in female, whereas they both are absent in male. Now the TRA2 gene, because it's an autosomal gene, and it is expressed in both male and female. So TRA2 protein is present in both male and female. Okay. If the TRA, so the TRA, so see, third line. So double sex gene produces pre double sex mRNA. We know that the gene will lead in case of eukaryotes. The gene is expressed in pre-mRNA. Okay. So if fourth line, if the TRA and TRA2 both are present, okay, so TRA is present in only female, okay, so now if TRA and TRA2 both are present, TRA2 present in male and female both, but the line says that if TRA and TRA2 protein both are present, the double sex transcript, that is the mRNA, pre-mRNA of double sex, is processed in a female specific manner and produces double sex F protein. F is for the female here. Whereas in the absence of TRA transformer protein, a male specific transcript is made which encode for DSXM protein. Okay, so what? So remember, so here we are talking about TRA protein and TRA2 protein. If they both are present, they will lead to transcription of double sex gene in double X. In a protein called DSXF, which will help in the formation of female, whereas because the TRA gene is not present in male, so in the absence of TRA, fifth line, in absence of TRA, a male specific transcript is made which encodes a DS. So DSXF represses the genes required for male development and activates female-specific genes. DSXF is present only in females because TRA and TRA2 both are present, and because of which a DSXF is produced, and which represses the genes which is required for male development and activates the female-specific genes. Ultimately, XX Drosophila develops into a female. DSXM inhibits the female traits and promotes male traits. Ultimately, the XY Drosophila develop into male. Now, I'm going to summarize this in a flow. This is the flow chart. So starting from here, the XXL gene. So pre-XXL gene is formed. This pre-XXL, XXL gene lead to the pre-XXL RNA. This pre-XXL RNA is alternatively spliced with the help of XXL protein. So you can see the arrow. You can see the arrow. So the early. So because of presence of early promoter, XXL gene is transcribed to form pre-XXL RNA. This form XXL mRNA and this lead to XXL protein. Further, this XXL protein will lead to alternative splicing of this pre-XXL RNA, which is found by maintenance promoters. The maintenance promoter later on lead to the transcription of XXL gene into pre-XXL RNA. This pre-XXL RNA is alternatively spliced by the already present XXL protein in case of females, and that lead to the XXL protein formation. Further, we know that this XXL protein lead to the alternative splicing pre-TRA RNA. So the pre-TRA RNA got alternatively spliced because of XXL protein which was present in females, and thereby TRA protein was formed. Because the because XXL protein, a fully functional XXL protein is present in females, thereby it lead to alternative splicing of pre-TRA mRNA in case of females. So TRA protein is only present in females. Now this uh, uh, pre-TRA2 RNA is formed in both male and female. So TRA2 protein is there. In female, the TRA is present, TRA2 both are present. Because of both are present, plus means because both are present, this both will lead to formation of they will lead to formation of a DSX mRNA in a form which will lead to formation of DSXF DSXF protein, which help in formation of female protein. This represses the genes for male development and further lead to an embryo develops as a female. So I guess I have explained you this process, and if you have any doubts, you can you can let me know, and we can discuss this. So this was the molecular mechanism behind the embryo, behind the sex determination in case of um, Drosophila. So to summarize that, in case of Drosophila, we have read that the Drosophila has four pair of chromosomes. So and in case of Drosophila, the sex chromosome, the XX, okay, and XY. So in case of Drosophila, XX, if there is present of X and X, so basically the ratio between sex chromosome and autosomal chromosome decides the fate of embryo. If the ratio is 1 or above 1, it will lead to formation of a female, and if it is 0.5 or less than 0.5, it will lead to formation of male, and if it or meta male, and if it is between 0.5 to 1, if it is between 0.5 to 1, it will lead to intersex. So the ratio of between the sex chromosome and autosome decides the sex of an embryo. In case of Drosophila, the Y chromosome do not decides the sex of a Drosophila, whereas the Y chromosome only decides that the male will be fertile or not. So if a male, so now a male with X naught, I mean Y is not there, so the formula is X naught. This means that the Drosophila is male, but the Drosophila is not fertile. But if it is XY, it means that the embryo will develop into into male, but it will be and it will be fertile. Later on, we discussed about the molecular mechanisms behind the Drosophila sex discrimination, and we majorly talked about four gene. So one was sex lethal gene, other was transformer gene, the third one was TRA2 gene, and last one was double sex gene. So there was an interplay. So there was a connection between four genes, and the so in case of female, the formation of one protein lead to formation of other, and second lead to formation of third, and third lead to formation of fourth and the fourth protein formed in a way that it will lead to the expression of those genes, which will help in the development of embryo into a female specific manner. So I guess and now I guess we have completed this topic, and this I want yeah. So one more thing. So in case of humans, there was two X and one and in case of females, and one X and one Y in case of males. And we know that for doses compensation, so because there was two X and one X and there was only one X in case of male, so one of the X chromosome in females get inactive to compensate the expression of X in case of humans. One X was inactive, so both X, so out of two X, one was inactive. So X inactivation is a process of doses compensation, or is a method which lead to the doses compensation in humans. Whereas in case of Drosophila, there is in case of male, it will be X naught or XY. Here, so what is interesting is that so female has two X and male has one X naught or XY. Here, a X chromosome works twice of the X chromosome or of two X chromosome. I mean, so I repeat here, one X chromosome of male works twice to compensate of double expression of X of female. So this is very important. Please mark these things, and if you have any doubt, we can cover this.